The overall goal of the following protocol is to examine the effects of microbial infection on chronic inflammation in vivo using a physiologically relevant animal model. This is achieved by orally infecting mice with Porphyromonas gingivalis to induce local and systemic chronic inflammation. MRI can then be utilized to measure the progression of systemic inflammation in live animals, while local inflammatory bone loss is assessed by micro-CT, and atherosclerotic plaques can be visualized by on-face lipid staining after sacrifice. Ultimately, differences in alveolar bone volume and the accumulation of atherosclerotic plaques between P. gingivalis infected mice and uninfected controls can be measured based on micro-CT analysis and on-face lipid staining, respectively.
The major advantage of our animal model is that it allows you to examine both host and pathogenic mechanisms involved in chronic inflammation, and specifically at sites distant from infection. So you can look at systemic sites as well as local sites of infection.
Using transgenic mice and defined bacterial mutants, this model can help identify key aspects of pathogenesis, such as host and microbial factors contributing to disease.
Although this method can provide insight into the specific host and pathogen mechanisms involved in inflammatory bone loss and atherosclerosis, it can also be adapted for other mouse models of disease, including rheumatoid arthritis, diabetes, and cancer.
This is the high-field MRI core facility for Boston University, and my postdoc fellow Ning is going to demonstrate aspects of mouse imaging.